Imagine you eat a bowl of salad greens with a drizzle of olive oil and lemon. You feel nothing special. No caffeine buzz, no sugar rush. But somewhere in that meal, a tiny yellow plant chemical is quietly changing how your cells talk to each other. It nudges inflammatory switches. It whispers to brain cells under stress. It even seems to mess with how your body handles fructose. You've eaten this compound your whole life. You've almost never heard its name. Researchers, though, are suddenly obsessed with it, not because it's magic, but because it keeps showing up at the crossroads of inflammation, metabolism, and brain health. So, what is this thing? Why are labs all over the world spending real money and time on a molecule you've probably never Googled? And here's the twist: some of the most intriguing data has nothing to do with vitamins or antioxidants. It's about a very specific enzyme that turns fructose into a metabolic stress signal. Why would a quiet plant pigment care about the way your body processes fruit sugar? I'm going to show you why the scientists are so interested in luteolin and what we actually know and where the hype outruns the human evidence. I'm Chris Mearns, and this is the fructose model. Let's start with a simple question. What is luteolin? Strip away the jargon. Luteolin is a small, yellow-colored plant compound. It lives in foods like celery, parsley, thyme, chamomile tea, green peppers, and some fruits. And if you've ever eaten a stir-fry with celery, sipped herbal tea, or loaded herbs onto roast vegetables, you've already met luteolin, you just didn't know its name. Chemists call it a flavone, which is a type of flavonoid. That family name matters because flavonoids tend to interact with enzymes and receptors that control inflammation and cell signaling. But here's the weird thing. Luteolin's not a vitamin. You don't get luteolin deficiency. You could go your whole life and no doctor would ever measure it in your blood. So why are scientists zooming in on this one out of thousands of plant chemicals? They didn't start with the salad. They started with the petri dish. When researchers exposed immune cells in the lab to inflammatory triggers and then added luteolin, they saw some of the same classic inflammation signals get turned down. It's like watching a smoke alarm that suddenly quiets itself while the pan is still smoking. Once that happened, the questions multiplied. If it calms inflammatory signaling in a dish, does that translate to actual tissues in animals? Could it reach the brain? Could it matter for conditions driven by chronic, low-grade inflammation? Animal and cell studies started stacking up. Luteolin showed less activation of microglia, the immune cells of the brain, less activation of mast cells, the first responders that release histamine, altered activity of NF-kappa beta, and other major inflammatory switchboards. On paper, this sounds almost too good, but here's the catch. Most of these early wins were not in humans. They were in rodents or in isolated cells with higher doses that you would ever get from a plate of food. So we need to be careful. A lab dish is not a living person trying to get through a stressful Tuesday. And yet, the signals were strong enough that researchers did not back off. They took the next step. They started asking a bigger mystery. If luteolin can dial down inflammatory chatter, what happens to the organs that are most vulnerable to chronic inflammation and metabolic stress? That's where metabolism and the brain move into the spotlight. Before we tie luteolin to fructose, picture something you might have felt. You go through a phase with a lot of sweet drinks or desserts. Nothing dramatic, just a bit more sugar, a bit less sleep, a bit more stress. Over time, you notice that you're more tired after meals. Your focus is fuzzier, and your belly feels thicker even if the scale hasn't gone crazy. This is where the story usually stays shallow. Too much sugar is bad. Less sugar is good. The fructose model looks at a deeper level. Inside your liver and some other tissues, there's an enzyme whose whole job is to grab fructose and snap a phosphate onto it. That first snap is called fructokinase. Think of it as a switch. Flip it on, and your cell commits to handling fructose fast. In humans and animals, chronic overactivation of the fructose switch is linked to higher uric acid, more fat in the liver, and signals that can make you hungrier and even more insulin resistant. So here's the wild counterintuitive idea. What if part of the benefit of certain plant compounds is not just antioxidant power, but their ability to lean on this fructose switch and tell it to chill out? That's where luteolin walks back onto the stage. Mechanistic work suggests that luteolin can interfere with the activity of fructokinase, specifically the KHKC form that's very highly active in the liver and kidneys. When fructokinase is dialed down in these models, several things happen. There's less rapid depletion of cellular energy. There's less production of uric acid, a byproduct that's tied to metabolic stress. And downstream, you see less of the survival mode pattern that drives fat storage, inflammation, and sometimes brain fog. Now, most of this is still preclinical. These are animal studies and cell experiments, and we do not yet have large, well-controlled human trials showing that luteolin stops fatty liver disease or reverses metabolic syndrome just by blocking fructokinase. But the pattern is really intriguing because it connects dots that used to feel separate. You can have a plant compound that calms inflammatory signaling in immune cells. The same compound shows neuroprotective effects in animal models of brain injury and neurodegeneration by toning down microglial activation and oxidative stress. And now you have evidence that it interferes with fructose's main entry into metabolism through fructokinase. Suddenly, luteolin is not just anti-inflammatory. It starts looking like a traffic cop standing at the key intersection between sugar handling, energy status, and inflammatory signaling. And if that model holds in humans, even partly, it could change how we think about the value of certain herbs and vegetables. Maybe about combinations of food patterns and targeted compounds for people under metabolic and cognitive stress. But what do we actually know in real people with all their chaos and complexity? Here's the direct answer to our starting mystery. Luteolin is a plant flavone found in everyday foods like celery, parsley, thyme, and chamomile. And researchers are studying it because it keeps showing up at the intersection of inflammation, metabolism, and brain health. And that alone changes the way we think about food. We usually divide food compounds into nutrients and antioxidants. Luteolin suggests there may be another category entirely. Think of it as a signal modulator. Instead of simply cleaning up damage after it happens, it appears to influence some of the switches that determine how our cells respond to stress, inflammation, and energy availability. Now, it's important to keep the evidence in perspective. The human data is still limited. We have encouraging pilot studies, strong animal research, and a growing body of mechanistic evidence, but we do not yet have the large clinical trials needed to make definitive medical claims. What we can say is this: researchers from many different fields keep arriving at the same pathway. Different diseases, different organs, different laboratories, yet they continue to converge on the same metabolic switch. If that pattern continues to hold, then the question isn't simply whether luteolin is beneficial. The bigger question is, what happens when we learn to safely turn down the survival program that fructose metabolism appears to activate? Because if chronic disease has a common denominator, it may not be inflammation, insulin, cholesterol, or calories alone. It may be cellular energy. And if cellular energy has a common upstream regulator, fructose metabolism is becoming one of the strongest candidates. If you're wondering why we formulated Sugar Shield around liposomal luteolin, now you know. The goal was never to create another antioxidant supplement. It was to target one of the most intriguing upstream pathways in metabolism using a compound with compelling mechanistic rationale and an encouraging, though still emerging, body of research. I'll leave links below if you'd like to explore that science in more detail. I'm Chris Mearns, and this is the Fructose Model.